Dear students, now we are going to discuss CAM metabolism, crossylation acid metabolism. It is an alternative method of dark reaction or carbon assimilation pathway. Actually, it is a variant of a C4 cycle. Crossylation acid metabolism. CAM. Crossylation acid metabolism. Okay, so this one is a alternative method. Of C3 and C4, but actually it is a variant of a C4 cycle. Okay, present in desert plants, plants which are growing in hot and tropical plants, plants which are growing in hot climatic condition, generally xerophytes. Example, cactus. Crossulaceae family. Okay, so they are with a C3 cycle. C3 cycle is common for all photoautotropes. This is a common method of a biosynthetic pathway that is common for all plants which are able to perform carbon reduction. They are also with a C3 cycle. Okay, so cactus, crossulace, and they are with a stomata. What type of stomata? Scotoactive stomata. Scotoactive stomata. Nighttime open, daytime closed. That is a type of a xerophytic adaptation in those plants. They are with a scotoactive stomata, right? Next, in CAM plants, for CAM plants, one kind of photosynthetic cell required, just mesophyll enough, like a C3 plants. Okay, so within one cell, for example, mesophyll here I'm taking, mesophyll with both C4 and C3 cycles, both are present. That's why I told you a variant of a C4 cycle. It is a variant of a C4 plant. This is an adaptation by means of which xerophytes, cactus xerophyte, are able to survive and able to perform photosynthesis in heliophytic conditions where the temperature is 40-45 degrees. Right? Next, in the case of C4 plants, C4 cycle, C3 cycle separated by space. Separated by space means two types of cells are involved. C3 cycle is restricted to bundle sheath cells only. And what about here? Within one kind of cell. But here, these two means a C4 cycle and C3 cycle. C4 and a C3 are separated by time, not the space. They are within same space, same location, but separated by time. Night time, daytime. So during night time, C4 cycle. During daytime, C3 cycle. We'll see what is that. Now, what about a mechanism of a CAM metabolism in the case of a cactus? So take one kind of a cell, one is enough, mesophyll cell. This much information was not given in both NCERT and Telugu Academic textbook. Just here I'm elaborating it. So this one is a mesophyll cell. So one kind of cell is enough. Now, what are the reactions within mesophyll cell? During night time, dark period time, night time, no sunlight, in the absence of sunlight. So separate these two so-called cell into two zones. Reactions in the absence of light, reactions in the presence of light. Okay. Daytime. Okay, so for example, this is a chloroplast. This is a granule chloroplast. Dummy diagram, granule chloroplast. Granule chloroplast. Here I'm writing. Granule chloroplast. Right? Now, so what about the reactions? So already within the chloroplast, starch is available. That starch is dissociated into triose phosphate, G3P. Triose phosphate, glyceraldehyde 3-phosphate. The triose phosphate is exported and converted into PEP, phosphoenol pyruvic acid. Okay, so now PEP accepting HCO3 minus. In the presence of a pepcase, phosphoenol pyruvic carboxylase. So in the presence of a pepcase, PEP accepting one molecule of HCO3. So product is a malic acid, wait, oxaloacetic acid, wait, oxaloacetic acid, OAA, oxaloacetic acid. So that oxaloacetic acid is converted into malic acid. Wait. So convert into malic acid. Same mechanism like a C4 cycle. Here malate dehydrogenase. malate dehydrogenase. Here take one molecule of NADH oxidized to one molecule of NAD plus. So OAA is reduced into malic acid, right? So now this malic acid is transported into vacuole. This is vacuole, SAF vacuole. Okay, so accumulation of malic acid. Malic acid accumulated. So malic acid is a organic acid. So during night time, opening of stomata and exchange of gases, CO2 is available during night time within the mesophyll cell. In order to control rate of transpiration to minimum, they developed this type of adaptation, having scotoactive stomata, opening of that stomata during night time, and uptake of CO2 like that. So during night time, a number of or whatever the concentration of malic acid increasing. So during night time, acidification means accumulation of a number of organic acids, particularly malic acid. So night time completed. Now daytime. What about during daytime? Now, so now malic acid is transported into chloroplast. Here I'm taking separate chloroplast. Chloroplast. Now malic acid is available within chloroplast. So malic acid. So now malic acid is decarboxylated. CO2 and pyruvic acid. And the CO2 is accepted by C3 cycle. To synthesize a glucose molecule. So pyruvic acid is converted into starch. So there is no regeneration of phosphoenol pyruvate or pyruvic acid, right? So this one is a sort of a C4 cycle. During night time, carbon is assimilated. Carbon is fixed in the form of acid. Same acid is subjected to breakdown, decarboxylation, and making CO2 available for C3 cycle to reduce that one into glucose molecule. So during daytime, stomata closed down, there is no exchange of gases. And what about rate of transpiration? Less. So during daytime, deacidification. Okay, so this much is not required. See how many kinds of cells involved? Only one kind. This one is a C4 mechanism. This one is C3 cycle. Both are taking place within one cell, but separated by nighttime, daytime. Separated by time only. This is one type of adaptation to survive in xeric climatic conditions. So that is enough for CAM metabolism. Crossylation acid metabolism. Okay, starch, triose phosphate, PEP, HCO3, oxaloacetic acid, reduced into malic acid, accumulation of malic acid within vacuole, nighttime completed, acidification. Now acids into chloroplast, where malic acid is broken down into pyruvic acid, CO2 is available for C3 cycle to produce glucose molecule. Here pyruvic acid is converted into starch. There is no regeneration of PEP, phosphoenol pyruvic acid. Okay, this is enough. For NETA, this much is not required. This is for MSET only. Now, photorespiration. Photorespiration. It is also called as a C2 cycle. This is very, very important for NEET, C2 cycle. And this is also called as a glycolate. Glycolate metabolism. Glycolate metabolism. So what is photorespiration? Light induced uptake of oxygen and evolution of CO2 by the chloroplast or chlorophylls is called as photorespiration. So it is a waste process. During photorespiration, fixed carbon is oxidized instead of reduction. So actually C3 cycle is called as C3 cycle is photosynthetic carbon reduction, PCR. C3 cycle is nothing but photosynthetic carbon reduction. What about this one? PCO, photosynthetic carbon oxidation. See the difference. Okay, C3 cycle is also called as photosynthetic carbon reduction pathway, biosynthetic pathway. So this one is a photosynthetic carbon oxidation pathway, oxidation cycle. It is a cyclic pathway, right? So here in C3 cycle, the critical step is, after understanding so-called one critical step, we'll go to what about the mechanism of photorespiration. So critical step in C3 cycle. Critical step of C3 cycle is carboxylation. Carboxylation reaction. Here the problem is a enzyme. That enzyme is a Rubisco. Ribulose 1,5 bisphosphate carboxylase oxygenase. So this enzyme is able to perform both carboxylation and oxidation, right? So for example, here I'm taking a Rubisco enzyme. Wait, RUBB. Ribulose 1,5 bisphosphate 5 carbon sugar, pentose sugar. Okay. So if CO2 is available, more amount of CO2. Now Rubisco role is carboxylation. So now carboxylation. And the synthesis of glucose molecule, and this one is called as C3 cycle. Okay, so if it is a CO2 is accepted by Rubisco, the pathway is called as a C3 cycle. Otherwise, if O2 is accepted, oxygen, so this one is definitely oxidation of carbon. Same enzyme is able to perform either carboxylation or oxidation. It depends on concentration gradient between CO2 and oxygen. If CO2 concentration is more than the oxygen concentration, definitely C3 cycle. If it is reverse, more O2, less CO2, oxidation. So this process is called as a C2 cycle. Okay, so for C3 cycle and C2 cycle, same enzyme. And RUBP, acceptor of CO2 common reactant. What are the uncommon reactants? CO2 and oxygen. If CO2 is involved, C3 cycle. If O2 is involved, C2 cycle. So during C3 cycle, glucose is produced. The product of carboxylation is definitely glucose molecule. During C2 cycle, no glucose molecule, no sugars. During C2 cycle, decarboxylation, carbon is oxidized into CO2. There is no formation of sugar molecules. So that's why it is a waste process. What about the rate of photosynthetic yield? Drops down because of deviation from C3 cycle to C2 cycle. So it is one example for competitive inhibition. So CO2 and O2 are competing for Rubisco. And ultimately, either CO2 or O2 is able to bind with Rubisco. It depends on the concentration gradient between CO2 and O2. Generally, Rubisco gives more priority, more importance to CO2 and less importance to oxygen. So if O2 is accepted by Rubisco to join that one with RUBP, there will be no C3 cycle, C2 cycle. There is no formation of glucose. Fixed carbon is oxidized into CO2. There is no formation of glucose molecule. So it is a deviation from C3 cycle to C2 or not? Right? Now, what about a mechanism? Right? Now, mechanism of photorespiration. So here, to execute a C2 cycle, total number of kinds of cell organelles required. 3 kinds of cell organelles. What are they? And what are the reactions taking place in them? First involved is chloroplast. First one is chloroplast. So chloroplast is a double membrane bonded cell organelle. Chloroplast. Right? So here, this is actually C3 cycle. Kelvin cycle, C3 cycle. Actually, this chloroplast have to perform C3 cycle. Okay, so if the concentration of CO2 is more than the concentration of oxygen, but under certain conditions, O2 concentration is more, CO2 concentration is very less. Under these conditions, now RUBP molecule, one molecule of RUBP. Here you can take two molecules of RUBP. Accepting how many oxygen molecules? 2 molecules of O2. 2 O2 accepted by RUBP in the presence of Rubisco enzyme. So here enzyme is adding oxygen to RUBP. RUBP is actually acceptor of CO2, but here RUBP is accepting oxygen. So this is oxygenation or not? Oxidation. So in the presence of Rubisco, at high concentration of oxygen, RUBP is oxidized into what are the immediate products? What are the products? So here, 2 types of products are produced. What are they? 2 molecules of phosphoglyceric acid. 2 molecules of phosphoglyceric acid. This is a 3-carbon compound. Next, 2 molecules of phosphoglycolate. Phosphoglycolate. It is a 2-carbon compound. Phosphoglycolate. Right? 3 carbon, 2 carbon. So 2 into 3, 6 carbons. 6 plus 4, 10. Here are 2 molecules, 2 into 5, 10 carbons. Right? So now, dephosphorylation, elimination of 2 inorganic phosphates from the phosphoglycolate. Now the product is a glycolate. Glycolate. Glycolate is a 2-carbon compound. This one also 2-carbon compound. Here enzyme required is a phosphatase. Enzyme is a phosphatase enzyme. Phosphatase enzyme. What about the cycle photorespiration? C2 cycle. Glycolate. Glycolate metabolism. Glycolate is the main substrate to run C2 cycle. Hence the name glycolate metabolism. Place of formation of glycolate is within the chloroplast. So now it is transported into next cell organelle, that is peroxisome, microbody, cell organelle, microcell organelle, microbody. So how many molecules? 2 molecules of glycolate transported into peroxisome. Peroxisome. Peroxisome is a single membrane bounded cell organelle, microbody. So now 2 molecules of glycolate. So now 2 molecules of glycolate is converted into 2 molecules of glyoxylate or glyoxylic acid. Here reaction is oxidation. Here 2O2 consumed. Okay, and the byproduct is 2H2O, hydrogen peroxide. So 2 molecules of glycolate is converted into glyoxylate or glyoxylic acid. Here enzyme required is oxidase. Oxidase enzyme is required. Right? So glycolate is oxidized into glyoxylate or glyoxylic acid. Here also 1 molecule of 2 molecules of O2 consumed. Next reaction is now 2 molecules of glyoxylate is converted into 2 molecules of glycine. 2 molecules of glycine. Glycine is an amino acid. Here amination is required. NH2 added. 2 amino groups required. Amino group, NH2, NH2. So 2 molecules of glyoxylate is converted into 2 molecules of glycine. Enzyme required is a transaminase. In the presence of transaminase, 2 amino groups are added to glyoxylates. 2 molecules of glyoxylate is transaminated into 2 molecules of glycine. Glycine is amino acid. Next, mitochondria. Next involved is mitochondria, double membrane bonded cell organelle. So now 2 molecules of glycine. 2 molecules of glycine. Glycine is an amino acid. Now, 2 molecules of glycine is converted into 1 molecule of serine. Serine is a 3-carbon compound. This one 2-carbon compound. Place of formation of a glycine is a peroxisome. Place of conversion of glycine into serine is a mitochondria. So what are the reactions here? Glycine is a deaminated, first deamination. That amino group is accepted here, deamination. Next, decarboxylation. 1 carbon is oxidized and removed in the form of CO2. So during photorespiration, carbon is converted into decarboxylated or removed in the form of CO2 within the mitochondria. Next, 1 molecule of NAD reduced into 1 molecule of NADH. Oxidative decarboxylation. Oxidative decarboxylation. So definitely they'll ask you, during photorespiration, CO2 is evolved within mitochondria. And what are the amino acids produced? Glycine and serine. Right? Now, 1 molecule of serine transported into peroxisome. Place of formation of serine is within mitochondria. Right? So now, deamination, removal of amino group. Actually, this amino group is coming from serine molecule. Deamination. In the presence of a transaminase enzyme, it is converted into hydroxypyruvate. Okay, so serine is deaminated into hydroxypyruvate. All these are 3-carbon compounds. 3-carbon compounds. Now, hydroxypyruvate is reduced into here 1 molecule of NADH is oxidized into 1 molecule of NAD plus. Hydroxypyruvate is reduced into 1 molecule of glycerate. Glycerate or glyceric acid, 3-carbon compound. Now, glycerate is available within the chloroplast. Glycerate. Now, glycerate is converted into phosphoglyceric acid. Here, 1 molecule of ATP is consumed. Right? And phosphoglyceric acid will enter into C3 cycle. Right? So here a very, very important thing is there is no formation of sugar. ATP consumed, NADH consumed without the formation of sugar molecules. There is no formation of sugar. Instead of that, the carbon present in REBP is decarboxylated. Okay, so it is a cyclic pathway started within chloroplast and completed within chloroplast. It is a cyclic pathway. What are the reactions? If the concentration of oxygen is more than the concentration of CO2, RUBP is accepting oxygen in the presence of rubisco. Products. 2 molecules of phosphoglyceric acid that will enter into C3 cycle. Okay, and 2 molecules of phosphoglycolate. Dephosphorylation of phosphoglycolate into glycolate. Glycolate is the precursor for C2 cycle. It is called as C2 cycle because the number of carbon atoms present in the precursor main substrate to run C2 cycle is a glycolate. Now glycolate is transferred into peroxisome. 2 molecules of glycolate is oxidized into 2 molecules of glyoxylate in the presence of oxidation enzyme, and hydrogen peroxide is produced. Now, two molecules of glyoxylate or glyoxylic acid is aminated to two molecules of glycine. Place of formation of glycine and amino acid is within peroxisome. So here, amino group 1, amino group 2. Two amino groups required. For each glyoxylate, one amino group. How many glycine? Two glycines. Now, glycine is transferred into mitochondria. Now, what about the fate of glycine? Glycine is deaminated, decarboxylized, and oxidized into serine. Here, one molecule of NADH is produced. Place of decarboxylation is within the mitochondria. Now, place of formation of serine is within mitochondria. Serine is transferred into peroxisome. What about the fate of serine? Deaminated into hydroxypyruvate. Now, hydroxypyruvate is reduced into glycerate. Here, one molecule of NADH is consumed. Now, glycerate is transferred into chloroplast. Now, glycerate is phosphorylated to PGA, phosphoglyceric acid, entering into C3 cycle. This is complete photorespiration. But in the case of a NCERT textbook, what is given? That is very, very important. Okay, right? So in NCERT, the given one is in the case of C3 cycle. For example, here I'm taking C3 cycle. If it is a C3 cycle, one molecule of RUBP accepting one molecule of CO2. Enzyme is same enzyme, Rubisco. Okay, so it is a 5 carbon. Okay, and this is a 1 carbon. Total number of carbon atoms, 5 plus 1, 2. So product is 2 molecules of PGA, phosphoglyceric acid. So if CO2 is binded to Rubisco, definitely the product is 2 molecules of phosphoglyceric acid. Go back to C3 cycle. Otherwise, deviation from that one, that is C2 cycle or photorespiration. In the case of photorespiration, this one is a photosynthetic carbon reduction pathway. This one, photosynthetic carbon oxidation pathway. See here, same RUBP reacting with oxygen. This is given in NCERT. RUBP, one oxygen molecule. In the presence of same enzyme Rubisco. How many carbon atoms? 5 carbon atoms. No carbon, only oxygen. So 5 carbon is cleaved into one molecule of 3-phosphoglyceric acid and one molecule of phosphoglycolate. This is very, very important. So this is a product 1 and a product 2. This is a 3 carbon. This is a 2 carbon. Right? So in the case of C3 cycle, REBP, CO2 in the presence of Rubisco into 2 molecules of phosphoglyceric acid. If it is photorespiration, instead of formation of 2 phosphoglyceric acids, REBP, by accepting oxygen, it is able to produce 1 molecule of 3-phosphoglyceric acid. But here it is a 2:1 ratio. Plus 1 molecule of phosphoglycolate. This was given in a textbook. During photorespiration, ATP consumed. During photorespiration, ATP and NADH, both are consumed. And without, there is no formation of sugars. And carbon is carbon oxidized to CO2. Fixed carbon is removed. Then what about rate of photosynthetic yield in C4 plants? Declines. Reason because of C2 cycle. But what about in the case of C4 plants? For example, here I'm taking C4 plants. In C4 plants, two types of photosynthetic cells are involved. This one is a C4 cycle. This one is C4 cycle. Go back to what I explained, C4 cycle. And this one is a C3 cycle. Always C4 cycle is able to increase CO2 levels within the bundle sheath cells. This one is mesophyll cell. As C4 cycle is continuously able to add or increase CO2 concentration, what about C3 cycle? It is not falling into photorespiration. So that's why in C4 plants, there is no photorespiration. Reason? C4 cycle is able to supply required concentration of CO2. It is able to maintain high CO2 concentration within the bundle sheath cells. But what about in the case of C3 plants? In C3 plants, there is no C4 cycle. The catalytic activity of rubisco is influenced by oxygen also. Here also, rubisco is present, but that is not influenced by oxygen. Reason? Always the concentration of CO2 is more than the concentration of O2 available there. So that's why in C4 plants, photorespiration is absent because of C4 cycle. If C4 cycle is stopped, if it is not working or not functioning, or there is no proper concentration of CO2, definitely, even in the case of C4 plants also, photorespiration is possible, right? So here, very, very important thing is that during photorespiration, how many kinds of cell organelles are involved? During photorespiration, or to complete photorespiration, 3 kinds of cell organelles required: chloroplast, peroxisome, mitochondria. Chloroplast, mitochondria, double membrane bonded cell organelles, semi-autonomous cell organelles with a DNA as the genetic material. And peroxisome, single membrane bonded cell organelle, microbody. And during photorespiration, the substrate, the main substrate to run the cycle is cyclic pathway, glycolate, 2-carbon compound. And what are the amino acids produced? 2 types of amino acids. First glycine, next serine. Place of formation of glycine is peroxisome. And place of formation of serine is within the mitochondria. Next, decarboxylation is within the mitochondria. During conversion of glycine into serine, 1 carbon out of 2 molecules of glycine, 1 carbon is oxidized into 1 molecule of CO2. So that is about photorespiration, right?